Usually when we discuss bacterial pathogens in C. elegans, we're looking at some form of chemical warfare.
Right, yeah, the classic toxin model.
Exactly. A bacterium secretes a toxic protein. That toxin binds to a target in the worm's intestine, and, a physiological cascade basically leads to death. It's an invisible molecular level attack.
Oh, absolutely. We map the receptors, we track the enzymatic degradation of the cell membranes, we trace the stress response pathways. It is almost always a story of chemical incompatibility.
But today, we're looking at an entirely different paradigm. What if the bacterial diet didn't poison the worm chemically, but instead waged an aggressive, purely physical war?
Yeah, mechanical destruction.
Exactly. We are talking about a scenario where the standard microbial food source secretly manufactures solid, commercial-grade bioplastics right inside the nematode's gut. Welcome back to the Deep Dive.
It's great to be back.
Now, before we jump in, if you want to help us uncover more of these incredible biological mysteries, please take a quick second to follow, drop a like, comment, and share this with your fellow nematode enthusiasts. It is truly the best way to help us grow this community.
Yes, please do. We really appreciate it.
So today our source material is an absolutely wild April 2026 PLOS biology paper. It's titled, Bacteria Producing the Bioplastic Polyhydroxybutyrate Kill the Nematode Caenorhabditis elegans.
It really is a remarkable piece of research. I mean, instead of looking at typical pathogenesis, we're basically tracing a microscopic murder mystery.
A murder mystery with plastic.
Right. We'll follow the mechanism from the moment a sudden environmental carbon spike forces these otherwise totally benign bacteria into, well, a metabolic hoarding state.
Which leads to the mechanical destruction.
Exactly, right down to the catastrophic physical collapse of the worm's digestive system. And perhaps most fascinating, we'll examine a deeply paradoxical genetic mutation that allows a fraction of the worms to actually survive this mechanical destruction.
I cannot wait to get into that mutation. But let's get straight into the metabolic setup first. So, as you know, we have the standard laboratory diets for the worms, right? Like E. coli, OP50, or HT115.
It's a standard menu. Yeah.
Right. But for this study, the researchers shifted the menu to Commomonas DA1877, which is just a common soil bacterium. And on its own, Commomonas is a perfectly viable food source.
Yeah, the developmental timing might shift slightly compared to OP50, but generally the worms are healthy.
Right, totally fine. The trouble only begins when the researchers supplement the agar with massive amounts of carbon. Specifically, they used 75 millimolar concentrations of lactate, or in parallel assays, they used pyruvate.
And that environmental context changes everything. I mean, when fed the Commomonas diet supplemented with that heavy hit of lactate, the vast majority of the worms experience severe L1 developmental arrest and then, well, lethality.
They just die.
Yeah. And the very small percentage that actually managed to push through to adulthood, they exhibited a really pronounced DAR phenotype.
Right, the deformed anal region phenotype, which is visually striking and obviously a clear indicator of severe intestinal distress or bacterial colonization. But okay, let's unpack this.
Sure.
My immediate thought reading the setup was just about direct toxicity. I mean, if you dump 75 millimolar lactate onto a plate, you have to wonder if the carbon source itself is inducing some kind of osmotic shock, right?
Oh, absolutely.
Or just acting as a direct toxin to the worms.
Yep.
It's like humans getting violently sick from eating way too much junk food in one sitting.
Well, the researchers actually anticipated that exact critique. It's a very fair point. So to isolate the variable of the bacterial metabolism, they utilized a paraformaldehyde assay.
Oh, to kill the bacteria.
Exactly. They took the Commomonas, treated it with paraformaldehyde to render the cells metabolically dead but structurally intact, and then they fed those dead bacteria to the worms on the exact same 75 millimolar lactate plates.
And the worms were completely unaffected.
Totally fine. The L1s developed normally. There was no DAR phenotype, no lethality whatsoever.
Wow, so the lactate isn't inherently toxic to the nematode at all.
Not at all. The toxicity is entirely dependent on the active internal metabolism of the Commomonas. The bacteria are, they're actively consuming that carbon surplus and converting it into a lethal payload.
Which forces the whole investigation directly into the bacterial genome, because now you have to find out what they're making.
Right.
So to uncover what that payload was, the team executed this massive transposon mutagenesis screen. They generated a library of I think it was approximately 5,500 distinct mutant strains of Commomonas.
That's right, about 5,500.
And they cultured all these strains in the high lactate environment, fed them to the worms, and basically just looked for any bacterial mutant that failed to trigger the lethality.
Yeah, it's a huge undertaking. And out of those 5,500, they isolated 56 mutant strains that actually allowed the worms to survive.
And this is where it gets crazy. When they mapped the insertion sites for those 56 rescued strains, a glaring pattern emerged. 6 of the specific genetic disruptions mapped directly to the biosynthesis pathway for polybeta-hydroxybutyrate, or PHB.
Right. A biodegradable bioplastic.
Which is just wild.
It is. And to understand why a bacterium is, manufacturing commercial-grade plastic, we have to look at the metabolic logic of Chamaemonas.
Okay, lay it on us.
When this organism encounters an environment incredibly rich in carbon, which is provided here by the lactate, but lacking commensurate levels of nitrogen or phosphorus, it can't actually utilize that carbon to divide and multiply. So it enters a state of physiological hoarding.
It just channels all that excess carbon into energy storage molecules.
Exactly. And it does this through the Phi A operon. So the bacterium takes 2 molecules of acetyl-CoA, condenses them using the Phi A gene product, reduces that intermediate via Phi B, and then finally the Phi A synthase polymerizes those monomers into these massive, dense, highly insoluble intracellular plastic granules.
Carbosomes.
Yes, carbonosomes. And the mutant screen showed that disrupting FACSI, so breaking that final polymerization step, completely rescued the worms.
It really is like swallowing a microscopic 3D printer. Under normal conditions, it's just printing, easily digestible biological material.
Right, totally normal food.
But the moment you flood the system with this raw carbon resin, the printer just goes rogue. It starts extruding solid plastic blocks directly into the worm's stomach.
That is a perfect analogy, and the researchers actually proved this wasn't just some weird artifact of Chamaemonas biology. They proved it through a brilliant synthetic biology approach.
Oh, right, the E. coli experiment.
Yes. They took our standard benign E. coli HT115. Now, naturally, E. coli does not possess the Phi beta operon. It totally lacks the genetic hardware to synthesize PHB.
Which is why a high lactate environment never turns HT115 lethal.
Exactly. But they basically engineered the hardware into it. They took the Phi operon from Chamaemonas and cloned it directly into the HT115. And crucially, they also added a gene called FAthP, which encodes a phasin protein.
And why is the phasin so important there?
Well, the phasin is critical because you can't just synthesize raw hydrophobic plastic inside a watery cytosol. It has to be packaged.
Oh, I see.
Phasin proteins coat the surface of the carbonosome, which stabilizes the granule within the bacterial cell. So by introducing the biosynthetic operon alongside the structural packaging protein, they fundamentally altered the metabolic capacity of a perfectly safe organism.
And the results of that engineering are just staggering. I mean, this modified E. coli, which is an organism that has been the backbone of C. elegans research for decades, suddenly starts churning out PHB carbonosomes.
Yep.
And the moment the worms ingest it, you see the exact same pathology. Immediate L1 arrest, profound lethality, and the DAR phenotype in the few survivors.
It's instant. The production of the bioplastic is entirely sufficient to trigger the collapse.
But exactly how does this plastic kill the worm? Because knowing the biochemical pathway is one thing, but witnessing the physiological destruction requires looking closer.
It really does. So the researchers employed transmission electron microscopy, or TEM, to visualize the mechanical load on the worm's anatomy. And the TEM images, they reveal a level of physical obstruction that is just difficult to overstate.
Because normally the intestinal lumen is just a relatively clear tube,
Yeah.
right? Like it's just processing liquefied bacterial soup.
Right. But in these worms, the lumen is massively distended. It is packed wall to wall with solid, highly electron-dense PHB granules. The gut is just entirely stuffed with plastic.
But the obstruction actually begins long before the intestine, right?
It does. C. elegans utilizes a specialized biomechanical structure in its pharynx, a grinder.
Right.
And its function is to crush intact bacterial cells. It disrupts their membrane so the intestine can actually absorb the intracellular contents.
But the material properties of polymerized beta-hydroxybutyrate are vastly different from a typical lipid bilayer. The worms are literally trying to crush solid plastic beads. It's like pouring liquid cement down a kitchen sink. As it solidifies, it doesn't just block the pipes. When you flip the switch for the garbage disposal, the sheer rigidity of the cement physically snaps the metal blades.
That is exactly what's happening. The researchers found that in roughly 80% of the affected worms, the pharyngeal grinder was visibly deformed or completely shattered by the mechanical stress of the plastic.
God, 80%. And then that biomechanical failure just cascades into a catastrophic neurological disruption, doesn't it?
Oh, it gets worse. We have to consider the defecation motor program, or the DMP. This is one of the most highly regulated, predictable physiological rhythms in the entire nematode.
Every 53.8 seconds,
A
right?
remarkably precise interval, yeah. The cycle is governed by a pacemaker in the posterior intestine. It initiates a rapid-fire intracellular calcium wave that propagates anteriorly.
And then triggers the muscles.
Right. This calcium transient coordinates the sequential muscle contractions required to clear the lumen. And the researchers actually utilized transgenic worms expressing fluorescent calcium sensors, GCAMP strains, to monitor these signaling waves in real time.
And what happens when the intestine is physically packed with carbonosomes?
That 53.8-second rhythm completely disintegrates. The calcium waves become highly erratic. I mean, sometimes the transient initiates in the completely wrong intestinal ring.
Wow.
Other times it fires but just fails to propagate down the length of the gut at all.
So the physical distension of the intestinal cells is likely just overwhelming the mechanoreceptors
Right.
and disrupting the ion channels required for that calcium cascade.
Exactly. And in the most severe cases of plastic impaction, the calcium waves simply cease. The entire motor program just shorts out, leaving the worm totally unable to clear even a fraction of the blockage.
So let me get this straight. The grinder is fractured, the lumen is packed solid, and the automated clearing mechanism has failed entirely. But the paper outlines one final structural failure that really illustrates the severity of this mechanical stress. The Smurf assay.
Ah, yes, the Smurf assay. It is such a vivid diagnostic tool for assessing intestinal barrier integrity.
So they basically feed the worms blue food dye, right?
Exactly, a non-absorbable blue food dye. In a wild-type healthy worm, the apical junctions of the intestinal epithelium maintain a strict paracellular barrier. So the dye remains entirely confined within the lumen.
It just craces the digestive tract.
Right, while the surrounding pseudocolome and tissues remain totally clear.
But when they ran the Smurf assay on the worms impacted with PHB, the blue dye didn't stay in the tube. It leaked directly into the intestinal cells and permeated the whole body cavity.
Yeah, it's brutal. The sheer physical outward pressure exerted by the accumulation of solid plastic didn't just cause a blockage, it physically ruptured the cellular barrier of the gut.
The worms are literally splitting apart at the cellular level from the inside out.
It is a systemic, multi-tissue collapse, and it's driven entirely by the mechanical properties of a microbial storage polymer.
Which brings us to the twist, because the second half of this paper is so compelling. The researchers didn't just stop at understanding how the worms die. They wanted to know if the host organism possessed any evolutionary countermeasure.
Right? Is there any hope for the worms?
So they shifted their focus from the bacterial genome to the nematode's genome, and they performed an EMS mutagenesis screen on the worms themselves.
Yeah, they mutagenize approximately 10,000 haploid C. elegans genomes. They subjected this vast library of mutant nematodes to the lethal PHB-producing Comomonas diet, basically searching for a genetic escape hatch.
And they actually found one. A single survivor strain emerged from that massive screen. This one specific mutation allowed roughly 50% of the worms to survive ingestion of the exact same plastic-laden bacteria that obliterated the wild-type population.
And through really rigorous whole genome sequencing and sibling subtraction techniques, they pinpointed the exact locus of the rescue. It was a singular point mutation in the NUC1 gene.
And this is where the biological logic of the paper takes a massive, totally counterintuitive turn.
It really does. What's fascinating here is what NUC1 actually is. It's a DNase II homolog.
Okay, break that down for us.
Well, its primary baseline function is to be secreted into the intestinal lumen to degrade the genomic DNA and RNA of the bacteria the worm consumes. Basically, it chops up the microbial genetic material so the worm can salvage the nucleotides.
So under normal physiological conditions, NUC1 is absolutely essential for efficient nutrient extraction.
Right. And by mutating it, you are actively crippling the worm's ability to digest bacterial DNA.
And that is the core paradox of this entire mechanism. I mean, the nematode is dying because its digestive tract is fatally obstructed by solid chunks of bioplastic. And the genetic mutation that rescues it from this mechanical death disables its ability to digest DNA.
It sounds absurd.
On a macroscopic level, that makes zero sense. If your home's plumbing is completely backed up with hardened resin, you don't solve the problem. by deliberately shoving massive clumps of hair down the drain.
Right.
You were just adding more raw material to an already fatal blockage.
I know, and the researchers acknowledge that exact paradox, and the physical reality in the gut actually confirms it.
Wait, really?
Yeah, when you image the NUC1 mutants feeding on the pathogenic bacteria, you can visually confirm that large accumulations of undigested, high molecular weight bacterial genomic DNA are physically piling up inside the lumen, right alongside the plastic granules.
So there is objectively more mass in the gut.
Objectively more mass, yes.
Yet the outcome is drastically improved. Because by utilizing fluorescently tagged carbonosomes, the researchers track the clearance rates. And in the wild-type worms, the plastic enters the pharynx, immediately aggregates, and just forms these permanent, immobile blockages.
But in the NUC1 mutants, the plastic granules remain dispersed. They just slide through the damaged grinder, they move through the intestine, and they are eventually excreted.
So the undigested DNA is actively facilitating the clearance of the plastic. How?
Well, the working hypothesis advanced by the author centers on altering the surface chemistry of the carbonosomes. We established earlier that the PHB inside the bacteria isn't naked plastic.
Right, it's densely coated in structural phasin proteins, specifically PAM and PHAR.
Exactly. And those specific bacterial proteins have a secondary binding affinity. They possess a very strong affinity for binding to DNA.
Oh, wow.
Right. So in the wild-type worm, NUC1 rapidly degrades the bacterial DNA into individual nucleotides, which leaves the phasin-coated plastic granules totally exposed.
And because PHB is highly hydrophobic, these exposed granules strongly repel water and instantly aggregate together.
Forming the massive rigid clumps that shatter the grinder and rupture the intestine.
But in the NUC1 mutant, the DNA isn't chopped up. Long, intact strands of bacterial genomic DNA just flood the intestinal lumen. And because those FAM and PHAR proteins on the surface of the plastic have a high affinity for DNA, the genomic strands rapidly bind to the outside of the carbonosomes.
And that DNA acts as a hydrophilic coating. By wrapping the highly hydrophobic plastic granules in a layer of water-attracting genomic DNA, the surface properties of the carbonosome are fundamentally inverted.
So they don't stick together.
Exactly. The tendency of the granules to aggregate and form rigid blockages is drastically reduced.
So it's basically acting as a biological laxative.
Pretty much, yeah.
The strands of DNA wrap the sticky, clumping plastic, reducing the friction and allowing the granules to just slide past one another. The worm is literally surviving a fatal physical obstruction by weaponizing the bacteria's own undigested genetic material against the surface proteins of the plastic.
It is incredible, and the researchers actually attempted to validate this biochemical interaction in vitro. They mixed purified PHB granules with high molecular weight bacterial DNA to see if it increased solubility in a test tube.
And did it?
Well, the in vitro results were somewhat ambiguous. Purified systems rarely capture the complex molecular crowding and, the precise protein coating ratios found inside a living bacterial cell.
Sure, that makes sense.
But the in vivo data is unequivocal. The physical accumulation of DNA in the NUC1 mutants definitively correlates with the disaggregation and clearance of the plastic load.
It is an unbelievably sophisticated host microbiome interaction. The nematode is exploiting a highly specific biochemical affinity between a bacterial structural protein and raw DNA, just to alter the material physics of a lethal polymer. It's brilliant.
It really is.
But, stepping back from the molecular mechanics, I think the most vital part of this paper is the ecological context, because it is very easy to view a study like this as a fascinating but ultimately artificial laboratory phenomenon.
Right, a petri dish trick.
Exactly. I mean, a 75 millimolar spike of pure lactate on an agar plate isn't exactly a natural environment.
But it is critical to contextualize this outside the lab. The researchers actually conducted a bioinformatics analysis of the natural microbiota associated with C. elegans in its native soil and rotting fruit environments.
And the numbers they pulled from that analysis really reframe the entire predator-prey dynamic here.
They discovered that 43% of the diverse bacterial species that C. elegans naturally encounters and relies upon for sustenance possess the facies synthase gene.
43%.
Yeah. Nearly half of the worm's natural food web has the latent genetic architecture required to synthesize PHB plastic.
Which means this isn't an isolated lab quirk at all. Out in the wild, soil chemistry is incredibly volatile. A piece of fruit drops from a tree and begins to ferment, or a sudden rainstorm alters the local nutrient gradients.
And that causes a massive localized spike in available carbon while nitrogen remains locked up.
Exactly, and in those moments of environmental flux, nearly half of the bacteria the nematode consumes will just autonomously shift into hoarding mode.
They will rapidly transcribe the fis operon, polymerize the excess carbon, and fill their cytosols with carbonosomes.
So the nematode is constantly navigating a microscopic minefield. Its primary food supply can dynamically weaponize itself based solely on the ambient carbon to nitrogen ratio.
It's wild. It entirely shifts how we view the evolutionary pressures acting on fundamental soil organisms. They aren't just evolving resistance to secreted toxins anymore.
Right.
They are constantly adapting to sudden, massive shifts in the mechanical properties of their food. The survival of these base-level organisms depends entirely on their ability to mechanically process and clear these polymers before the physical load ruptures their digestive tracts.
And the NEC1 mutation we discussed may just be one of many complex physiological workarounds. These nematodes have evolved to survive in an environment where their food periodically just turns into solid plastic.
It really highlights how dynamic the soil ecosystem is.
And that reality brings up a much larger, honestly somewhat unsettling thought to leave you with today. Throughout human industry, we constantly praise biosynthesized polymers like
Oh,
polybeta-hydroxybutyrate.
totally. We label PHB as the ultimate green alternative.
Right, a biodegradable bioplastic that will solve our petroleum-based pollution crisis.
Yeah.
But if these natural bioplastics inherently act as lethal, weaponized, physical gut blockages for fundamental base-level soil organisms like nematodes, what happens to the mechanical stability of the global food web when we start mass-producing and dumping millions of tons of this eco-friendly plastic directly into the environment?
It definitely forces a severe reevaluation of how we define a material as biologically safe. I mean, biodegradable does not necessarily mean harmless during the degradation process.
It absolutely doesn't, and that is something to seriously mull over until our next deep dive. As always, thank you for joining us today, and a final reminder to like, comment, and share if you enjoyed this. Keep questioning the details, and we'll see you next time.